Hi, everyone. Thank you for joining today. We will get started in 5 minutes. Hello, everyone, and thank you for joining today. Welcome to the NOAA Omics Seminar Series. I'm your host, Nicole Miller, and I'm a NOAA Omics specialist. I sit in NOAA Ocean Exploration. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. The seminars from this series are made available on YouTube and posted to our Omics website. There should be a link to the website in the chat now. As participants, you are muted, but feel free to type questions into the questions for staff box throughout the presentation. At the end, I will read the questions for the presenter to answer. So with that, our presentation today is titled Pairing eDNA with an Inshore Acoustic Survey for Atlantic Herring, Challenges and Some Promising Findings by Dr. Graham Sherwood. I'm going to go ahead and share permissions to Dr. Sherwood while we get started.
Great. How's that, Nicole?
Let me take a look. Great. Perfect.
You're seeing that?
Yes. Yep. Real quick. Dr. Sherwood is a fisheries ecologist whose research at GMRI focuses on the intersections between food web ecology, behavior ecology, and energetics of diadromous, estuarine, coastal, and continental shelf fish species, primarily in the Gulf of Maine region. He uses a broad range of observational methods to discern patterns in movement, stock structure, feeding, growth, and reproduction of a broad range of commercially and ecologically important species in the Northwest Atlantic. This includes fisheries acoustics, tagging, stable isotope analysis, and various stock discriminating techniques like morphometrics and otolith microchemistry. More recently, his work has focused on pairing traditional sampling techniques with environmental DNA for the purpose of validating the latter as a fisheries assessment tool. Thank you, Dr. Sherwood, for presenting today. And I will now pass it off to you.
Great. Thank you, Nicole. And thank you for organizing this series. And also thanks to everyone who's tuning in. I just want to start out by acknowledging my co-authors. So it's myself and Aaron Whitman, who are at the Gulf of Maine Research Institute in Portland, Maine. And then we are collaborating with Mike Kinnison and Geneva York at the University of Maine in Orono. And so Aaron and me are kind of the muddy boots ecologists, the fisheries ecologists, and Mike and Geneva keep us honest with the lab work and the eDNA side of things. So just to get started, Nicole already mentioned sort of the background of the omics program, and so I won't go into too much of that, but basically why eDNA and why this work. And so eDNA is part of a larger NOAA strategy to apply 21st century technologies to benefit various sectors of the blue economy, including fisheries. So that's what I've highlighted here, but also aquaculture development, food and water safety, species and habitat conservation. And so with respect to fisheries, where can eDNA fit in most in fisheries discussions is with respect to informing fisheries surveys and ultimately assessments. And one of the questions is, can eDNA replace or at least complement traditional fisheries surveys? So thinking about trawl surveys is one of the more common techniques used for fisheries assessments. Can we somehow supplement some of that information? And it's actually quite timely given the fact that we know that there are some issues with trawl surveys. And just to give you an example of that, just imagine that you have a heterogeneous bottom. And so on the left, you have maybe a seamount rising up from the depths. And on the right, you might have some kind of gully. In both cases, you have very high relief structure. Superimpose on that a population of fish. And in this case, at high abundance, those fish might utilize all those habitats, including all of the space in between, which is sort of more uniform flat bottom habitat. So if you're designing a trawl survey, you're applying mostly a random stratified type design where you're randomly selecting sites, mostly stratified by depth, but mostly where you go ends up being where you're actually able to pull the trawl. So you're not going to get hung up on rocks, and you're not pulling a trawl down the side of a steep cliff, underwater cliff. And so in this case, at high abundance, you might actually get, a good depiction of the population. However, if the population were to change in some way and occupy some of those high relief habitats, then that same trawl survey would have a hard time actually capturing those fish. It would give you a different answer in terms of the actual abundance that's out there. This is in fact one of the issues we're having with cod in the Gulf of Maine. The survey says that there are actually not that many cod, which is true from the large scale perspective, but a lot of fishermen are actually finding cod in some of these structured habitats in what they call the footprint of the fishery. So the issue here then is, can we apply eDNA to maybe supplement some of this information, get into some of those habitats where we can't trawl? And so to do that, we need to actually validate eDNA as a useful method that actually gives us more or less the same answer as some of these more traditional. And so here's one of the earlier examples of comparing trawl sampling to eDNA. This is from Thompson et al. 2016, and this is a multi-species example from Greenland. And on the left, you can see where they have the number of trawl detections of various species across the entire study, and then that's regressed against the eDNA detections. And so you can see a positive relationship, but quite a lot of scatter around that relationship. And on the right, you can actually see what that translates to in terms of abundance and biomass in the red and green versus eDNA copy reads in the blue. For a single species comparison, we have this example from Salter et al. In 2019. This is Atlantic cod in the Faroe Islands. And on the left, what we see is sort of a more of a regional comparison, where if we're considering comparisons, at large scales, we do tend to get these nice relationships where in a particular region, if you see lots of cod in a trawl or a series of trawls, you're going to see a lot of eDNA. In areas where you don't see a lot of cod in the series of trawls, you're not going to see much eDNA. When you break it down by trawl, it tends to get a little less clear. There is somewhat of a positive relationship, but there are instances in this case where you see lots of eDNA and less trawl captures. This is another example comparing eDNA to another fisheries fisheries survey technique, and that is acoustics. And I believe this was actually a seminar last year, perhaps the year before, from Shelton et al. In this paper is 2022. It's considering Pacific hake on the US West Coast. And so here we have a large-scale acoustic survey where they are estimating acoustic biomass and then comparing that to the DNA results. And so you see this nice positive relationship, which is nice, but again, there's some scatter around this relationship. So the previous examples are promising, but is it possible that the robustness of the eDNA versus traditional survey relationships are even better than they seem? So a meta-analysis by Yates et al. In 2019 suggests that this might be the case. So when they looked at studies from the literature, and so those three that I just showed you were examples of studies that they might have considered in this type of analysis. When you compare those relationships in nature, where we have some kind of capture technique versus the eDNA relationship, those correlation coefficients in our squared values are lower than if you have some kind of a priori knowledge of what the actual animal or organismal abundance was going into the comparison. And so those are the laboratory comparisons. And so it's quite possible that in some cases we're underestimating the power of these comparisons just because of the fact that we may not be doing as good of a job as we think on the traditional side of the equation. So this would be true if catchability was low. So going back to the Thompson example with the multi-species comparison in the trawls, here again we have abundance in biomass of various species or families, and then the eDNA relation, the eDNA results. And in this particular case right here, there's lots of eDNA, but very little abundance, but some biomass. So it's quite possible that you know, one Greenland shark can put out a lot of DNA, and in fact, they did catch, I guess, some or one. I'm not sure what that biomass refers to. But again, in certain cases, you might get an instance where catchability might be low for certain species that might be putting off a lot of DNA. Another example on this is the second blue bar from the right. That's Paralepididae. I think those are barracudinas. Those are very skinny fish, and so we see the DNA from those, but those likely just swim through the net. So again, a catchability issue that could hamper some of our ability to derive relationships between capture and eDNA. So how to compare eDNA to a known quantity in the field? And I put known in quotations because it's never quite known, but can we get closer to this known quantity? And this is where this current project comes in. And this was the original title that was in the proposal to the Omics program. And that is an in situ test of the robustness of the eDNA for assessing fish abundance in an open marine system. Into pairing eDNA and acoustic survey for Atlantic herring. So this draws on a previous study that we conducted between the years of 2012 and 2016. So in this case, we partnered with 10 lobster fishers up and down the coast of Maine. And so each of them had 4 of these transects to go out and sample for acoustics over the course of the herring spawning season, which takes place in the fall in Maine. That is basically end of August to about the beginning of November. The herring also come into this, these red lines basically encompass the 90-meter isobath. So herring come into this relatively shallow area to spawn during this period. And so one of the things that we were able to derive from this survey over these 5 years was looking at things like spawning dynamics. So this figure shows, again, the coast of Maine. And then each of these dots is the location of a spawning school of herring. And one of the ways that we were able to derive that was based on sort of acoustics as well as behavior of herring. So in this case, on the right here, we have a herring school that's up in the water column. And then on the bottom, we have a herring school that's down on the bottom. All of these surveys took place at night to take advantage of the behavior that during the night, herring form these spawning schools near the bottom. And so what we did is we actually enumerated these schools by just considering those schools that were within the bottom quartile of the water column, and then mapping those and then coming up with this heat map approach here to show where spawning was more prevalent than other areas. And so what we see is 2 general areas that light up. One is just in the bottom there, that's right off of Portland or Cape Elizabeth. And the other one in the middle is right off of Penobscot Bay. Those red ellipses are a previous survey done in the 80s and 90s to look at herring egg deposition. And so herring are bottom spawners. They lay their eggs on the bottom in large mats. And so researchers in the past went out and actually surveyed the presence of those eggs, and those ellipses are the location of those eggs. And so it was encouraging to see that our acoustic results looking at the actual spawning behavior matched with those previous egg surveys. So again, this is one of the areas that we've seen to have very high spawning activity, lots of spawning schools right off of Cape Elizabeth. And I'm going to focus on this area for the rest of the talk. So this is what our survey looks like for the current study. And so this survey was conducted in 2020 and 2021 right off of Portland. And you can see the scale there in terms of the spacing of these acoustic transects and the spacing of the water stations. The acoustic transects are the white lines and the black dots are the water sampling stations. Each of those water sampling stations included 3 samples, one at the surface, one at the bottom, and then one in between surface and bottom, so a mid-water sample. We conducted the survey on the GMRI research vessel, the Merlin, which has a pole-mounted Simrad EK60 split beam system. So we're running 38, 120, and 200 kilohertz transducers system, which allows us to do some species partitioning based on the different frequencies. And I'll get into that in a little bit. One thing I should mention is that the previous survey, because it was working with 10 lobster boats, we actually distributed systems among those boats, and those were commercial grade systems, but nonetheless, we were able to collect valuable data from those systems. What we're working with here is a scientific echo sounder system. So this is an example of, a fairly definable herring school that was surveyed along that period that we initially were conducting the survey. So that was between 2012 and 2016. So one of the things that we wanted to do, in addition to pairing those acoustic transects with the water sampling station, so that's more of a spatial temporal kind of comparison, we also wanted to take advantage of the fact that in the past we could identify these schools and then we could sample from the schools to try to understand eDNA dynamics in relation to a school. So for example, if we sample in a school, we should expect to see lots of DNA. Maybe if we sample below it, we would see less. If we sampled above it, we might see less. And certainly if we sampled away from it, we might see less. And then we could also perhaps consider things like current direction, whether or not this sample on the left here is upstream or downstream of the school. So that was the plan. And so this is an example of what we were calling an opportunistic survey. So we had the systematic surveys which ran over those acoustic transects, and then we also went out for a number of nights to actually find schools and see if we could actually take samples in relation to those schools. So this is an example of that. We're going along, we encounter a school, we stop. Now we send down the Niskin bottle to collect the deep sample here. And what you're seeing here is the school's not big now, it's just that we're on top of it. So we're continuing to ping on it. Then we take a mid sample, we take a surface sample. This all takes about 10 minutes. And then we resume the survey, and then we actually note this school near the bottom. This is possibly the same school that just perhaps swam to the bottom, perhaps on its own, or just as a in relation to our disturbance. So nonetheless, we were able to sample from a number of these schools, and acoustically, just at first glance, they seem like herring schools. And so we wanted to test that. And this is where we run into our first challenge that was listed in the abstract and the title. Those schools that we encountered, all of them were not herring. So the question is, why are they not herring, and what are they? So to answer the first question, why aren't they herring? This is a time series of spawning stock biomass and recruitment for herring for the Northeast herring stock complex. This includes the Gulf of Maine. And you can see that when we conducted our 2012 and 2016 survey, we were in a period of relatively high abundance. The stock was actually doing relatively well. It was near the target biomass. It was maybe declining at the later years, but nonetheless, during those periods, we were seeing lots of herring that was related to a relatively healthy stock. You can see the recruitment was actually dropping off. That's the panel on the right. We conducted our survey at this period, well after historic low biomass values, as well as historically low recruitment. And so, unfortunately, we were working in a period where very low overall herring biomass, and then likely that hampered our schools and carry out the research that we had proposed. So getting to the second question, what are the schools? So we initially developed the qPCR primers for herring and tested these against a suite of co-occurring species for specificity. And this is work, again, that was done by Mike and Geneva at UMaine. And so the schools from the previous slide were tested for herring, and again, these came back negative. Subsequent tests revealed that the schools were not made up of any of the other fish species for which primers were developed. These included things like sturgeon, cod, and striped bass. We wouldn't expect those to be those schools anyway. They're not really schooling species, and they tend to be more, in the case of sturgeon, perhaps more coastal than where we were operating. But these particular species right here could easily form schools in the area that we are working in. And so we tested against those species, and again, all of those. A little bit of a head scratcher. One thing that prompted us to do was look a little closer at the acoustic signal. And this is where I think observer bias comes into play a little bit. Based on our previous experience, those schools that I showed, we did confirm that those were within the acoustic parameters of swim bladder fish, such as herring. And so when we were encountering similar size schools, schools with similar morphology, we were just sort of at first, we were thinking those are probably herring. But then when we look closer, we started to see some discrepancies. And so this is just an example of the two frequencies that we use, the 38 and the 120 kilohertz. And what's interesting that pops out in this region right here is those are the target strengths of what herring should be in the around the minus 45 decibel range. That's where you tend to see herring. This is the whole target strength frequency distribution for those two frequencies. And what you see here is that that first smaller peak there that's highlighted shows up on the 38, but not on the 120. And this is where we get into a little more into the details of acoustic analysis, but this basically draws on differential frequency responses of different organisms in the ocean, various frequencies. And so for a swim bladder fish like herring, it shouldn't matter too much whether you're operating at 38 or 120. You should see that similar target strength. And so we should have seen those peaks on both frequencies. But something like squid, for instance, has a differential response. And so we would tend to see it more on the 38 than on the 120. And that's the red line right here is squid. And the blue line I'm referring to is basically flat across those frequencies. And so this started, we started thinking then at this point that perhaps what we're looking at is squid. And so another reason that it could be squid is that squid are actually increasing in abundance in the Gulf of Maine. This is a response to the rapid warming of the Gulf of Maine. They tend to be more sort of mid-Atlantic. In years prior, they weren't as abundant in the Gulf of Maine, but so it could be a flip of the system where we're moving from a herring-dominated system to perhaps a squid-dominated system. And so that was the thought. And so we developed primers for both short fin and long fin squid. And both of those came back negative. And so we're left scratching our head, wondering what these schools are. We did do some metabarcoding. We initially ran an 18S run, and this was applied to gain insights on the invertebrate community within the schools. These samples came back dominated by tunicates. There wasn't really any indication of squid. And then we're currently running a 12S run for fish beyond what's already been tested. So these results are still out, and we're waiting to hear back. So those are to be determined. So that's where we're at with the school identification, and then using that as a means to compare to DNA. One of the things that I think really sort of pops out for me anyway for this result is that I think DNA gives us a really important way to ground truth schools. I mean, at some point we're going to figure out what that signal was, and so as opposed to doing things like net tows through these observable schools, if we can get a Niskin bottle in the school itself, we should be able to do a lot with ground truthing for acoustics in the future. So moving on, as I mentioned, we did the sort of more systematic surveys, and so I'm going to focus on those results now. And so this is basically just the combined tracks of all of our surveys in 2020 and 2021 off of Portland or off of Cape Elizabeth. We had two sets of transects, so we can only complete one set in one given night. But we basically ran over these about, I can't remember if it was 8 times a year or something on that order. I think there were 17 overall survey sorties. You see some squiggly lines in there, and those are when we were trying to track down schools and do some of the school work I was referring to. And then you can see, like, for instance, on this bottom track here, that's just running over the same transect repeatedly. And so this is just a depiction of what we partitioned as fish signal from the acoustics over all of those dates, over both years and all of the sampling dates. And then the NASC here, those are the bubbles. NASC refers to nautical area scattering coefficient, and that's basically a proxy for biomass. And so this is the biomass of what we think is swim bladder fish, and historically that would have been herring, but at this point we're not exactly sure if it's herring or not. But nonetheless, you can see some patterns. We tend to see much more biomass in the southern or the bottom left corner, and those are the sets off of Cape Elizabeth. So we're going to focus on those, on this set of transects right here. And so if we look at just, this is in one particular year, this is 2021, we can look among dates and we can look for some variations. So we're looking at both spatial and variation in NASC. Again, this is NASC that we're attributing to fish, possibly herring. We can see that there are some patterns across these dates that encompass the spawning time for herring in coastal Maine. And so some of the patterns are that NASC appears to peak on October 20th. This is consistent with our previous study from 2012 to 2016. On and after October 20th, NASC appears to be concentrated in the eastern or deeper side of the transects. And so that's highlighted by these red blobs right here. And I'll come back to those. So the question is, does eDNA track these patterns? So the good news is that we can detect herring. And so these are the results from all of those transects among those different dates. I'm just highlighting 4 here, and the 3 different depths. So you can see that we had a fair number of detections, and you can see the relative strength of those detections in terms of bubble size. The not so good news is that eDNA doesn't seem to follow the acoustic patterns, at least at this scale and apparent low biomass. And so if we superimpose those blobs from October 20th on, where we saw most of that acoustic biomass, there doesn't seem to be any real correspondence between the DNA results and the acoustics. I highlight November 4th right here on the bottom. We can actually see all of the DNA herring, or the herring DNA reads were away from that blob of higher abundance. In some cases, we did see some correspondence, but again, nothing to really draw any major conclusions. There was a depth dependency, so this was interesting. We saw 4 times as many detections on the bottom as we did on the surface. So this is, I think, interesting result for moving forward, just understanding the dynamics of DNA for herring. And I've mentioned that herring are bottom spawners, and so this could reflect some of that behavior. It could also reflect perhaps juvenile herring on the bottom, perhaps not even forming schools yet. One of the things about this bottom result, where we're seeing most of our detections, is that we decided maybe we're not looking at the right scale. So let's partition the acoustic signal to that bottom portion of the water column. And so in this case, what we did is we calculated NASC in the 10 meters off the bottom, and then looked at those same dates and those same locations. The gray bubbles show the distribution of NASC that we're attributing to fish, possibly herring, and then the red circles are the eDNA results. And again, the red circles represent the relative size of copy numbers results. And so again, not much of a correspondence here. So not really much to conclude from this at this scale and at this level of abundance. One of the things I should mention is that it's possible that our herring results are influenced by herring bait. We're working in an area that's very highly fished from the perspective of the lobster industry. And so historically, that would have included literally hundreds of thousands of traps baited with herring on the bottom. And so that could certainly influence our results. One of the things that might work in our favor is the fact that because the herring stock collapsed, the lobster industry adapted and moved on to different bait sources, such as redfish and even some rawhide. And so that shouldn't be an issue. We talked to some some fishers, and they said that in that area, if they were operating there in those years, 2020 and 21, they were not using herring bait. So that it remains to be seen whether herring bait is an actual issue, and that would be an interesting study to see how herring bait traps actually emit herring DNA. And again, I mentioned that some of this could be due to juvenile herring near the bottom that maybe not forming schools, that would create our DNA result, and then the schools that we're seeing in terms of the NASC could be some other fish species. All right. So one last nugget, I didn't mention, but when we developed primer sets for herring, we developed 3 different sets to target 3 different length amplicons. That is 150, 342, and 792 base pairs. And so all of the results that I've shown you so far are for the 150 base pair fragment. And this should be the more stable fragment and hence more readily observable. And so that's why we used it. We wanted to make sure that we were starting off on the right foot to get a result in that sense. And this follows from some work by Joe et al. In 2017 in Japan. And so this was a really interesting study where they were working with jack mackerel in an embayment. And they started off with the right panel right here, looking at the decay of different length fragments for jack mackerel. And you can see the blue here is our short fragment, and that's decaying much slower than the red fragment. And then when you actually compare the distribution of DNA using those different fragments, you get different results in comparison to the acoustic results. And so the more orangey colors right here in the middle, these are the acoustic results showing patchy distribution of mackerel, which is what you would expect for a schooling species. And then if you apply the short primer set, you don't see that patchiness. You see basically, that mackerel DNA inhabiting the whole bay, particularly at the surface. I should mention that that red area right there near the bottom it's a fish processing plant. But if you do apply the long fragment that decays quicker, you do see the patchy distribution, and that distribution more or less matches the type of patchy distribution that you see from the acoustics. So this really, I think, gets to sort of the importance of considering fragment size and then how we interpret eDNA data. So if we're really interested in the here and now, where a school is forming, potentially we're interested in spawning dynamics, where schools form and when, we might use something like the long fragment to get at that kind of information. If we're interested in long-term averages over larger scales, perhaps we would go with the short fragment. And so this was really interesting information. We wanted to see if we could actually apply that to our system. And so we conducted a similar tank experiment to look at decay rates of these 3 different fragments for herring. This is actually a recent run. Our initial run that we did a couple of years ago didn't quite pan out for various reasons. And so we took another stab at it, and this is what we just basically got hot off the press. And so you can see that here that first of all, the shorter fragment starts out much higher in terms of copy number, and then stays active over a much longer period of time, whereas 2 longer fragments start out lower, and then they become non-detectable, in this case, after 30 minutes, but we suspect it's probably longer than that. It's just that for this initial run, the next time point after 30 minutes was 4 hours. So it's likely that those fragments were still detectable to 1 or 2 hours. And so we have those data and we're going to analyze those. But nonetheless, we got very similar results to Joe et al. And this is something that we could potentially apply to our system, and we did. And so going back to our result that I showed you, the bubble plot of the detections short fragment in the black, you can see then superimposed on that is the detections for the longest fragment in the red. You can see that we get much fewer detections. I actually, that's a typo there, 46 for the red, I think it's 6 detections. So much fewer detections. Again, no real conclusions to draw in terms of comparing this to acoustic biomass, although Anecdotally, there was a very large school that was noted right here on November 9th at the mid-depth. And so this mid-water DNA that's showing up quite strongly here in this region right here could be related to that. That's exciting, but again, it's not enough to really draw any major conclusions. So just to wrap up and some take-homes. First is that eDNA is showing promise as a fisheries assessment tool across multiple studies. This study attempted to add to this body of knowledge by comparing eDNA to herring acoustic signals. Unfortunately, the Northeast Atlantic herring stock complex collapsed prior to the start of this study, making comparisons difficult. I think I've learned my lesson that it's probably better to study fish that are already in a collapsed state as opposed to fish that might actually enter a collapsed state. Nonetheless, it's encouraging that eDNA can detect herring at low biomass levels in our study area, and our results confirm the importance of considering eDNA fragment length when interpreting eDNA data. So I'll stop there. I'll thank you. Thank you to the program organizers. Thank you to funding from the Omics program, and also to SINAR, which is the Cooperative Institute for the North Atlantic region, which GMRI is part of for funding to come to GMRI. And then biggest acknowledgement here is our captain, Zach Whitener, who kept us safe at night. As I mentioned, these surveys were done at night to take advantage of the spawning behavior. And so that was a tall order for a captain to be out there all night conducting these surveys for us. So with that, I'll thank you all and take any questions.
Graham, thank you so much for your great presentation today. You may want to keep your slides up just in case we want to reference them during some questions. And we'll move forward with questions. We have one so far. Everybody, please feel free to post any in the questions box located in the GoToWebinar app on your screen. So our first question is from Mark, and Mark asks, how fast do herring schools swim? I'm wondering how likely you think it is that eDNA remains localized to where the schools are detected.
That's a really good question. And, you know, I think that there's definitely capacity for herring to reach all of the within the footprint of our survey. It's not that large when you compare it to that example that I showed you on the West Coast for Pacific hake. I mean, that was large scale. And I think that's where we're seeing like the best results in terms of the large scale comparisons. And so something like herring that could move around within the scope of our study could have obfuscated some of the results. With that said, in our 2012 to 2016 survey, we did see some level of site fidelity for spawning. And that is because herring will spawn over only particular bottoms. It's basically sort of more uniform gravelly bottom that basically enhances egg survival. And so if we had entered into this with a healthy herring stock and a healthy spawning stock, we would have expected the herring to actually hold bottom during the spawning period in certain areas versus others. And so in that case, we would have expected to see some more interesting spatial temporal results. But it's definitely a really good question. And I think that's where the fragment length also comes into play, in that, you know, if we're talking, you know, decay beyond detection within an hour for the longest fragment, then we're going to be less open to actually detecting those schools away from where they actually formed. And so that could be used to our advantage, whereas the shorter fragment might just give us a depiction of just herring over the entire area that we surveyed. But yeah, great question.
Awesome, great question, Mark. Our next question is from Mike, who asked, did you try generic markers that will tell you what species it is instead of targeted markers?
So yes, I believe that's, so that this is where the 12S run, I think, can come into play, and that one is currently out, waiting for results on that. So I assume you're referring to metabarcoding. And so, yeah, that's kind of where we're at right now. And I'm not sure if Geneva is on the line and is as registered as a participant, but that's basically my sort of understanding of where we're at with that.
Awesome. Our next question is by Danielle. And Danielle asks, great talk and interesting stuff. Is it possible that herring were the species schooling, but the eDNA collection was flawed in some way? How did you handle water after the Niskin sampling? Perhaps the pore size? And if you know it's something else, did you consider American shad as being a schooling species?
Yeah, great questions. And to be honest, I mean, that was one of our worries too, is that, you know, were we missing something? And we kind of reasoned that if we can detect herring at all, and we're putting a Niskin bottle right in the center of the school, we actually have echograms showing the location of the Niskin bottle right in the school itself. And so that should increase our chances of actually capturing some of the DNA. And so when we weren't seeing the DNA, there was a little bit of concern there. I think if Geneva's on the line, she could talk more about some of the quality controls. But nonetheless, we ended up using the same, the same approach, actually detecting DNA in other areas where there weren't schools. And so we think that the specificity of the qPCR is high enough that it can actually detect herring when they're around. And so that lends us to think that, we're not dealing with those schools being herring. And again, going back to some of that acoustic analysis, in retrospect, we should have known that those weren't herring schools when you look more closely at the actual acoustics.
Thanks, Graham. Our next question is from Annie, who asks, you showed that eDNA of herring was detected more towards the bottom of the surface. Does eDNA break down faster, closer to the surface, perhaps due to sunlight, surface turbulence, etc.?
Yeah, no, that's a really good question. And to be honest, I'm not really sure. I will say that these samples were all collected at night. And so to the extent that we're dealing with a time frame, starting at basically, sunset through the rest of the night, that's encompassing, 12 hours. We wouldn't expect sunlight to affect that signal at all, because again, those samples were collected at night. I honestly, I mean, it could be related to, circulation dynamics and stratification, and also school location. And so if these, if the herring are schooling near the bottom, it does make a lot of sense that we would see them on the bottom. And the herring bait thing also sort of makes a little bit of sense as well, if the fishers were using herring. And so I think it has a lot to do with, just sort of school location or fish location, as well as perhaps stratification.
Thanks, Graham. And tacking on to that question, I have a question. So do you think the disconnect between where we're seeing the fish acoustically and the eDNA could be related to geophysical dynamics at all, like turbidity or currents?
Yeah. So again, what you're saying is that where we're seeing the DNA could be just reflective of like some upstream effects.
Our next question is
sorry, that was a question to you, Nicole.
Mm-hmm.
I was trying to understand your question more. I hadn't really fully answered it.
Yeah. Yeah. So just kind of proposing if geophysical dynamics like turbidity or currents might and likely do influence how DNA moves throughout an ecosystem. And
I think those are all really good questions, and I think those are areas that we need to, dig into a little more. I think in terms of the disconnect, I'm pretty, at this point, thinking that a lot of those schools that we're seeing, I mean, some of them match up. And so when we do the acoustics, we're going to see herring, and we're going to see other species as well. And so that doesn't lend itself to very satisfying comparisons when we're focusing on the single species, such as herring for the eDNA. And the reason that it wasn't a concern when we started this study is because the herring signal overwhelmed everything else, anything else that could have been in the water column during the spawning season. And we just didn't see those spawners. And so at this point, we're kind of still left shaking our heads and trying to figure out, okay, we've got that acoustic biomass there. It doesn't match up with the DNA. And so what's going on is likely that acoustic biomass is reflecting other species, and that the DNA itself, where it's not matching with acoustic biomass, could be related to some of those dynamics that you're speaking of in terms of like currents, and also the fact that perhaps those species, the herring themselves might be closer to the bottom and not detectable. But yeah, definitely, an outstanding question for sure.
Yes. Well, we're looking forward to your future research. Our next question is from Madalena, who asks, thank you for the very interesting talk. Regarding the length of the target fragments you chose, do all markers target the same gene, or was qPCR used for both short and long fragments to quantify detections?
That is a Geneva question. And again, I'm not sure I don't know if Geneva joined us. I'm not sure if you can see that, Nicole.
Not yet.
That could be a question. If you want to contact us after, if Geneva's not here, then I can point that question in her direction.
Awesome. We do have a few more questions, Graham, so we'll continue on. Sure. Our next question is from Mike. Could you speculate more about what the mystery biomass detected by acoustics is?
Yeah, no, that's a really good question. And someone I think mentioned shad, and so I'm not sure that we ran qPCR for shad. I'm really interested. I was hoping to have that 12S run available for today, because I suspect that will help us elucidate what the signal is. But again, in some of the cases, not all of the cases, I only showed one of those schools that kind of showed that we're dealing with likely an invertebrate because of the differential frequency response. And so it's possible that it's made up of some kind of invertebrate. Typically, when we see invertebrate signal, though, it forms layers and not schools, other than things like larger invertebrates like squid. And so I would be surprised if those schools with very sort of definable boundaries are invertebrate in nature if they're not squid. And so the question then becomes, what are those schools? And we're really, it's just, it's been too long trying to figure out, what they are. And so hopefully we'll get some insight onto that in the next coming weeks.
Awesome. So we have a few more questions to move through, and then we'll end at the top of the hour. This one comes from Mark, who says, I can't remember if butterfish was the species you tested for, but they lose their swim bladder early in autogeny, and many have different signals at different frequencies, and may have different signal at different frequencies.
Yeah. That's actually really good. Yes. In fact, we have seen incursions of butterfish more and more into the Gulf of Maine. And so I think that is a very likely candidate. So that'll be something that we hope to perhaps get a little better information on. Yeah, for sure.
Our next question is from Jeff, who asks and comments, great talk, thanks for sharing. Just wondering about the turnaround time from collecting the samples to getting the eDNA results. Also, did you observe any problems with potential contamination?
Yeah, so in terms of the turnaround, we spent a lot of time trying to figure out, strategize how to analyze these samples. And so when we got hung up on the school identification, I mean, that was fairly quick. We collected those samples, and then we were able to get that negative result in a very timely manner. And then it was a matter of trying to come up with a strategy for sampling, given the fact that we knew that we were in a different kind of situation than when we had initially thought that we would get into in terms of higher herring biomass. So I think that the turnaround can be quite quick. I mean, you know, the experiment that I showed at the end there with the decay experiment, that was a within week turnaround for those qPCR results. And then contamination. I mean, I don't think so. I don't think it's a problem. I'd be interested to hear if other people have had issues with, you know, contamination. I mean, we apply, you know, to all of the sort of clean approaches that we can. You know, we sterilize the bottles. We do field washes. You know, we filter everything in a clean lab. And so, you know, I don't suspect that's an issue, and I don't think there's a lot of reason to expect that it's an issue, given the fact that we're just having more of a hard time actually detecting the herring than anything else. But yeah, I mean, those are all definitely concerns.
Thanks, Graham. Our next question is from Lenny, who asks, can you provide more information about the pore size and the filters used for capturing eDNA? And can you share the average concentration of eDNA per collection or filter? And this might be a question for Geneva.
Yeah, yeah, definitely.
Alrighty.
I don't know off the top of my head what the pore size was, but it was definitely sort of standard for this kind of work. That basically, working with Mike and Geneva, they recommended. But that's definitely, I think, a question that if you want some follow-up, we can point that in Geneva's direction.
Mm-hmm. Our next question is from Daniel, who asks, there are 3 different fragment lengths. Or are there 3 different fragment lengths, all from the same region of the genome? Were they mitochondrial?
I believe so, but again, Geneva did the primer development, so that would be a question for her. But that is my understanding, that I think they all were from the same region.
Okay. A few more questions here. One from Annie. Do you have a sense for how long eDNA of herring lasts in the water? I'm guessing it's dependent on water conditions. And we might have covered this briefly before, but.
Yeah, and that's sort of what we're trying to get a better handle on. And I think that some of our experiment that showed the decay of the different length fragments sort of gets at that. So that experiment was run, it was juvenile herring that went into it was a cooler, basically. It was like a 80-liter cooler with 2 juvenile herring, about 1.5 grams each. And so it was actually surprising that they let off that much DNA. They were captured, you know, a couple weeks ago in nearshore waters. So the water temperature was 17 degrees Celsius, and that was the temperature of the experiment over the entire decay period. So that's a little bit warmer than we would expect the herring when they're occupying the offshore region. So when we're dealing with spawning schools, we're getting closer to 10 degrees Celsius or lower. And so we would expect that the decay rate would likely be a little bit slower in that case. So I think it depends on a lot of factors. So there's going to be temperature. There's someone earlier mentioned potentially sunlight at the surface. And then of course, fragment length is a big one. And so I think that's kind of the $64,000 question. Is how long does DNA persist in the environment? And if we can sort of get to a point where we don't exactly know the absolute answer, but we can kind of bracket it in terms of understanding what a long decay period looks like versus a short decay period looks like, and then addressing our questions around that information, then I think we can gain a lot of insight.
Awesome. Well, Graham, thank you so much for your time today. Thank you, everybody, for joining. That concludes our question section. Thank you, everybody, for joining today. The NOAA Omics Seminar Series takes place on the 3rd Wednesday of each month. And you can find out more about past seminars and future seminars on the NOAA Omics website. and NOAA Science Seminar Series websites. Thank you all very so much, and special thanks to Dr. Graham Sherwood for your time today.
Thank you, Nicole, and thank you for all the great questions.
Alrighty, bye everyone.
Bye.